We are now moving on to our next unit, lecture unit 3, starting with chapter 6, which is about bones and bone structure. So your goals after this PowerPoint would be to be able to describe the functions of the skeletal system, classify bones according to their shape and internal organization, describe the major types of bone markings, and that is going to be more helpful for the lab portion of your course. So there is a slide in here on bone markings, but you definitely want to learn those terms for lab, but they won't be on your lecture exam. To identify the different types of cells in the bone and list their functions and relationship, to talk about the components of bone matrix and what the functions of the different components are, and also be able to describe what would happen if each component had a pathology. So if we had a disease affecting one component of the matrix, what would we see in that individual skeleton? And we'll learn about some diseases that relate to that. To compare the structure and location of compact bone and spongy bone, and discuss the structures of an osteon. We saw an osteon in our tissues unit, so we're going to revisit that, including some of the structures within. To differentiate periosteum and endosteum, as well as red and yellow bone marrow, and to compare endochondral ossification and intramembranous ossification, describe ossification, osteogenesis, osteolysis, and calcification, and then to discuss the epiphyseal cartilage and identify how it relates to growth of the bones, and what's the significance of the epiphyseal line. So the functions of our skeletal system. Our skeletal system is going to consist of the bones of our skeleton and also the cartilage, ligaments, and other connective tissues that hold it together. So our primary functions are going to be support. The skeleton provides a framework, basically the frame for our whole body, an attachment site for all those soft tissues. It's also a place where we store minerals and lipids. The bone itself is a reserve of minerals. Remember, we had those calcium salts that make up the solid matrix of bone tissue, osseous tissue. And those reserves help maintain concentrations of calcium and phosphate ions in our body fluids. So they're kind of a storage reservoir. And then yellow bone marrow actually stores lipids. Another function is blood cell production. White blood cells, red blood cells, and other blood elements are produced in the red bone marrow. The skeleton also protects our organs and is a leverage site for muscle attachment. It allows for movements of muscles in the body itself. If our muscles weren't attached to our bones, they would just be flopping around by themselves. There are 206 major bones in the adult skeleton, and those bones are classified by their shape and their structure. Bone shapes are divided into 6 major categories: sutural, irregular, short, flat, long, and sesamoid. So our sutural bones, also known as wormian bones, these are small, flat, oddly shaped bones, kind of like little puzzle pieces. And we find these in between the flat bones of the skull, which are the larger bones of the skull. And they're little. They can be as small as a grain of sand, sometimes as big as a quarter. And the borders are irregular, like a puzzle piece. Then we have the flat bones that I just mentioned of the skull. And the flat bones, they have these thin parallel surfaces, and we'll look more closely at that when we look at bones up close, but you'll see here there's thin parallel surfaces of compact bone within the flat bones. And we find these in the roof of the skull, as we mentioned, as well as in the sternum and the ribs and the scapula. They're good at protecting underlying structures and also sites for muscle attachment. Then we have irregular bones. So our irregular bones, these have complex structures with a variety of surfaces. They might have areas that are flat, notched, or ridged. And we find these examples of this type of bone is the vertebrae and the spinal column, the pelvic bone, and a few other bones within the skull. Long bones, whoops, erased instead of circled. Long bones are long and slender, and we find these in the arm, the forearm, the thigh, leg, the palms of our hands, soles of our feet, fingers, and toes. The femur is an example of a long bone, and it's the heaviest bone in our body. Short bones have a boxy appearance. The carpal and tarsal bones, so that's the wrist and ankle bones, are examples. And then we also have sesamoid bones, our final type. And sesamoid bones are small and round and flat, and they're found near joints of the knee, hands, and feet. And the number of sesamoid bones can actually vary from person to person, so we all don't have the exact same number of bones in our body. But everyone has one sesamoid bone, which is the patella, your kneecap. So everybody's got that sesamoid bone. Our bones not only have unique shapes, but they also have characteristic bone markings or surface features. And these can include projections, which are protrusions, where a muscle, ligament, or tendon can attach, or an articulation with another bone can occur. And we also have openings and depressions, which are basically holes or valleys where vessels or nerves are going to kind of lie against the bone or penetrate through the bone itself. And these markings can help us glean information about the bone and the individuals. For example, anthropologists and pathologists can determine the size of an individual, their age, their gender, and the general appearance based on skeletal remains. If any of you were fans of the TV show Bones, I don't know if it's still on or not, but I watched it a very long time ago. They kind of gave a Hollywood version of how anthropologists can actually find a skull and make a generalized image of what that person may have looked like based on the surface features, those bone markings of the skull. They had some really fancy CGI computer that did it. I don't know if something that fancy is actually real, but give you the idea. And so you should review these in lab, and then there is also a homework assignment going over these bone marking terminologies. But we have projections, so things that are sticking out of the skull. We have processes, which is a projection or bump. And then ramus is a part of a bone that forms an angle. We have the ramus of the mandible or jawbone. And then different openings that we have within the skull. We have sinuses, which is an air-filled chamber. Foramen is a circular passage for blood vessels or nerves. A fissure is more of a slit going through the bone. A meatus is a passage or channel. And then a canal is basically a channel or passageway through the bone. The meatus is kind of the opening of the channel. Other terms, depressions, including a sulcus, which is a narrow groove, a fossa is a shallow depression, places where we might have muscle or ligament attachment. A trochanter is a large rough projection. A crest is a prominent ridge. A spine is a pointy process. A line is a lower ridge, so big ridge would be a crest, low ridge would be a spine. A tubercle is kind of the smaller version of a trochanter, so it's a smaller rounded projection. A tuberosity is more of a roughened projection, a smaller roughened projection. A head is an expanded articular end, so it's usually a large round end of one of the long bones. A neck is kind of a narrow connection between the head and the diaphysis, which we'll learn those terms in a minute. A facet is a small flat articular surface, so another part of the articulation, the joint. Condyles are smooth, rounded articular processes. And then a trochlea is a smooth, grooved articular process. When we look at the structures of our bones, we have a variation in the structure of long bones versus flat bones in the body. The long bone can be divided into 3 parts. The diaphysis is the shaft, the long skinny part in the middle of the bone. Whoops. And it has an outer wall of compact bone. You can see here on either side it's got this outer wall of compact bone, which is dense and solid and so very protective. And then inside that compact bone wall within the diaphysis we have the medullary cavity running between those 2 walls of compact bone, and that's where the bone marrow lives, within the diaphysis. The epiphysis is the wide part at the end of the long bone, so this and this. And it's made mainly of spongy bone, also called trabecular bone. It has an open network that looks like a lattice. Unfortunately, we can't see each other in lab to pass one of these bones around, but it almost looks like a sponge, which is why it's called spongy bone. Like a sponge that has been turned into bone. So it has kind of a lacy appearance within that spongy bone of the epiphysis. And then the metaphysis is basically that area where epiphysis and there's fighting hummingbirds behind me, where the epiphysis and the diaphysis meet. There's my cursor. So that was long bones. We also have flat bones, and those flat bones, remember we saw these in the skull, the ribs, and the sternum, and they have a little bit of a different structure. They're basically like a sandwich of spongy bone, where the bread is our compact bone here on either side, and that's referred to as the cortex. And then the meat, or peanut butter, depending on your type of sandwich, in between those 2 slices of compact bone bread, is made up of spongy bone, as we saw in the epiphysis of the long bone, and it's sometimes called the diplo. The flat bone does not have a big medullary cavity like the long bone does. There is red bone marrow present in the spongy bone, though. So just a little review. I want you to quiz yourself here, because we did talk about osseous tissue or bone tissue already. So what kind of tissue of bone, what category does it fall into in our 4 major tissue categories? So it's a supporting connective tissue. Remember, bone and cartilage are supportive connective tissues. The matrix of the bone is dense due to what salts? We had calcium salts are what make that bone matrix dense. In addition to those hard salts, we need a type of fiber to provide some flexibility. The analogy we talked about previously was that the calcium salts were like the concrete in our structure, and then we had these reinforcing steel beams, and that was our collagen fibers. And then the type of bone cells found within this matrix are osteocytes. So those osteocytes, remember, they lied inside little holes or pockets, just like the chondrocytes. And those holes or pockets are called lacunae. And then we have those little narrow passageways through the matrix that allow gas and nutrient and waste exchange from those osteocytes. So remember, we had that osteocyte lying within that little lacunae, and then it had these little passageways, little extensions through the bony matrix where its little cytoplasmic extensions could reach out, and those were called canaliculi. And then the outer surface of the bone, except within the joints, is called periosteum. So let's talk about that bony matrix. The bone matrix is made of calcium phosphate. That accounts for 2/3 of the bone mass. And it interacts with calcium hydroxide to form these hydroxyapatite crystals. It also incorporates other calcium salts such as calcium carbonate and ions like magnesium. And this part of the bone matrix is a part that's very hard but brittle. So it's able to withstand compression but isn't able to twist or bend. So this is the part of the bone matrix that you would think of like a thick eggshell. And then we have the other part of the matrix is our collagen, and that accounts for 1/3 of the mass. And the collagen has the tensile strength, allows for some twisting and bending. For proper function, we need both parts of the matrix, but we sometimes can have diseases that affect one or the other. So take a moment to think to yourself about what could happen to bones if one component of the matrix was missing, if we were missing the calcium phosphate component, or if we were missing the collagen component, what we might see. And this image on the right-hand side is a little experiment you can do at home. Normally I bring some bones into lab that I've done this with, but you can kind of simulate what would happen with these diseases. If you have some leftover chicken bones from cooking, you can take one bone and you can put it into a container of vinegar for several days. And the vinegar will actually remove the calcium phosphate portion of the bone. And what you'll be left behind with is the matrix without the calcium, but with those collagen fibers. And then you end up with this bendy bone here. It's pretty impressive. So if you have time, throw a chicken bone into a cup of vinegar and see what happens after a week. To simulate what it would be like without the collagen fibers, you can take the bone and bake it for an extended period of time. The results aren't quite as impressive as the removal of the calcium, but it will give you a little bit of an idea of how kind of light and brittle the bone can become. So bone cells, we talked about the matrix. Remember, tissue is made up of, connective tissue is made up of a matrix with cells lying within that matrix. So the bone cells make up only 2% of the bone mass. And we have 4 types of bone cells actually: osteogenic cells, osteoblasts, osteocytes, and osteoclasts. So our osteogenic cells, which you can see here, our osteogenic cells, or osteoprogenitor cells, are stem cells. Specifically, they're mesenchymal cells that divide to produce osteoblasts. And we find these in the inner cellular layer of the periosteum and in the endosteum, which lines the medullary cavity. So we said before we talked about the fact that we have that periosteum, that is surrounding the outer edge of the bone, but we also have what's called an endosteum, which is a similar layer that lines the medullary cavity. So these osteogenic cells are found in these layers. And these cells maintain the osteoblast population, and they also assist in fracture repair. Next, we have our osteoblasts. So remember, those osteogenic cells help make osteoblasts. These osteoblasts are immature cells, and they produce new bone matrix in a process called osteogenesis, or ossification. Remember, at the beginning of the semester, we talked about the fact that if we have cells that end in blast, it often means they're an immature cell, and a cell that ends in cyte is the adult cell, or the mature cell. So these osteoblasts make and release proteins and components of the bony matrix, and that matrix is called osteoid before it's become calcified. So they make this osteoid first, and then the osteoblasts help increase the local amount of calcium phosphate, and that triggers the deposition of calcium salts in the matrix, and that converts that osteoid to bone. Osteoblasts surrounded by bone matrix then become osteocytes. So we've got our osteogenic cells, which turn into osteoblasts, which create osteoid, which then gets calcified, creating our bony matrix. And then that osteoblast is surrounded by that mature bony matrix and becomes an osteocyte. So our osteocytes are the mature bones in the solid matrix, and those are the ones that we could see in lab when they were the little black ovals when we were looking at the tissue in the microscope slides. And osteocytes do not further divide. They are the adult cell. The osteocytes, remember, lie in a lacunae, that little pocket between the layers of the matrix. In bone, there's only one osteocyte per lacunae, unlike cartilage where we can have more than one chondrocyte lying within there. And remember, we have those little cytoplasmic extensions coming off of the osteocyte. So we've got little arms coming out, and the little canals that they're going through, the little passageways, are called our canaliculi. And that allows the cells to communicate with each other and also allows them to access nutrients and get rid of wastes. These cells have gap junctions, which allow cells to exchange ions and small molecules. And we also have diffusion occurring through the interstitial fluid surrounding the cells and their little cytoplasmic extensions. The two major functions of osteocytes include maintaining the protein and mineral content of the matrix. Our bone matrix is continually being broken down and rebuilt. And the osteocytes dissolve the matrix, and that allows the minerals to enter circulation when needed. Then the osteocytes rebuild the matrix, stimulating new hydroxyapatite crystals to be deposited. So that process of breaking down and rebuilding bone is constantly going on. The osteocytes also help repair damaged bone. If an osteocyte gets released from its lacunae, they can turn into a less specialized cell like an osteoblast and then can assist in bone repair. And then our final bone are osteoclasts. Our osteoclasts absorb and remove bone matrix. They look a bit different than the other cells we looked at because they're large multinucleate cells. They can have 50 or more nuclei. What other cell did we talk about that had lots of nuclei? Think back, quiz yourself on your tissues unit. Skeletal muscle, remember? So these osteoclasts are found in shallow depressions within the bony matrix called osteoclastic crypts. And basically these crypts have kind of eroded into the bony matrix here, into those osteoclastic crypts. And the osteoclast secretes acids and protein digestive enzymes that dissolve that bony matrix and can release the minerals that are stored there. These cells have little finger-like processes, which are called a ruffled border. And you can kind of appreciate that in this picture, how there's little processes, little edge there in the image. And much like microvilli, this ruffled border is going to increase the surface area that's in contact with the bony matrix. The erosion process is called osteolysis or resorption. And the released products are then resorbed at this border. This helps regulate calcium and phosphate ion concentrations in body fluids, so it's very important in homeostasis, and we'll talk more about that later. One thing to note about the osteoclasts is that unlike the osteoblasts and osteocytes, they are not derived from osteogenic cells. They are derived from the same stem cell that produces monocytes and macrophages. So they have a different progenitor cell than the osteoblasts and osteocytes. Remember, with the other cells we talked about, we start with the osteogenic cell, then we transform to an osteoblast, and then to an osteoclast. So they are all related. These sorry, I think I just said osteoclasts. So with our other cell line, we start with the osteogenic cell, which then turns into the osteoblast, which then turns into an osteocyte. Remember, blast immature, cyte adult. These osteoclasts are not within that line of cells. They have their own progenitor cell. So in our compact bone, remember that's the bone that made up the walls of the long bone or the bread of the flat bone sandwich. So in our compact bone, we have what's called the basic functional unit. The main component of that compact bone at a microscopic level is called an osteon, or sometimes called a Haversian system. Sorry, my dog's running around out here. You can probably see his tail behind me. So osteocytes within this osteon are arranged in these concentric layers around a central canal, which has one or more blood vessels. And these blood vessels carry blood to and from the osteon. That central canal is usually running parallel to the surface of the bone. Get my little drawing tool. So each of these circles here is an osteon. So the compact bone is made up of multiple osteons. Here they've just kind of pulled it up out of the compact bone. And so we've got these rings, these concentric rings within where the osteocytes are arranged. And then the central canal, which runs through the center of each osteon. And that central canal is usually parallel to the surface of the bone. And then running perpendicular to the bone surface, we have what are called perforating canals, or Volkmann's canals. And these passages carry blood to the osteons deeper within the bone and also to marrow in the medullary cavity. The bone matrix itself forms layers called lamellae. And these lamellae are arranged in those concentric layers around that central canal. So they're like the rings on a tree or an onion when you cut that osteon and you see those rings. Sometimes people say they look like a target and the central canal is a bullseye. And then each lamellae contains collagen fibers that add strength. We also have interstitial lamellae that fill the spaces between the osteons and circumferential lamellae that are found at the outer and inner surfaces and are produced during bone growth. So if we're looking at, erase this, if we're looking at the different lamellae, shush, we've got our concentric lamellae are the rings that make up each osteon. Those are our concentric. Then we've got our interstitial. Remember, interstitial kind of means the in-between areas. Interstitial fluid is the fluid outside of our cells or in between our cells. Interstitial lamellae is the lamellae kind of between the osteons. And then we've got our circumferential lamellae around the outer circumference of the bone. So I recommend drawing yourself a little cross-section of compact bone so that you get an idea of how those lamellae are arranged and know the different terms. In the long bone, the osteons are aligned in a parallel fashion. So they're aligned parallel to the surface, or to the axis, along the long axis of that long bone. And that makes the bone strong when stressed along the axis of alignment. You can kind of think about your bone as a straw. If you had like a drinking straw standing up and you, I hate this camera, and you pushed the straw on either end. I wish I had a straw here to show you. It would be very hard to bend it. But then if you took the straw and turned it sideways, it doesn't have much strength in that direction because you're pushing against that axis of the straw, or in our analogy, the osteon. And this affects how bones can be injured, how they can be fractured. And this is just a close-up of the osteon, or the structure of the compact bone. So we've got our osteons, our lamellae, central canal running through each perforating canal, running perpendicular, our little osteocytes lying within lacunae, communicating with their little caniculi, extensions. And then we've got concentric lamellae, interstitial in between, and circumferential on the outside. And these are just images from microscopes of the osteons, which we looked at in the tissue unit. Sorry, I had to let my dog inside, but I got a straw while I was in there. So what I was trying to say in those last slides is imagine our osteons kind of arranged parallel to the surface of the long bone. If you try and push on your straw in this direction, you really cannot bend it. It has a lot of strength in this direction, and that's kind of where we would want this strength. Think about our long bones. Most of the pressure would be coming from this way as it supports our body. But if we bend in this direction, it's very easy to bend the bone. So sometimes when we get an abnormal force upon our bone where we're acting against that long axis of the bone, we can get a fracture. Okay, so spongy bone. We were just talking about compact bone. Spongy bone, remember we see that in the epiphysis and in the flat bones. It makes up the meat or the peanut butter of your sandwich between those layers of compact bone. So in spongy bone, we don't see the osteons that we see in compact bone. The matrix of spongy bone forms a meshwork of fiber bundles that are called trabeculae. And it's kind of this branching open network. You can see them here. So these are those trabeculae, and that's what gives it this spongy appearance with these little branches of matrix going in different directions. And those trabeculae are oriented along stress lines, and they have little cross braces for support. The spongy bone does not have capillaries or veins within the matrix. Remember, our compact bone, we had those vessels going through the central and perpendicular canals. We don't have that within spongy bone. Instead, in the epiphysis of long bones and the interior of large bones like the sternum and ilium, we have red bone marrow, and that's what forms blood cells. Blood vessels within the marrow deliver nutrients to osteocytes in the spongy bone by diffusion across canaliculi that open onto the surface of trabeculae, and you can see those as little holes here in our picture. So we've got little caniculi, little channels through the spongy bone, and here's our little osteocytes lying here. So their little extensions are gonna extend out to the edge of the trabeculae and then be able to communicate with that red bone marrow and get their nutrients that way, and also get rid of their wastes. The framework of the trabeculae protects the cells of the marrow. Yellow bone marrow is also found in other sites of the spongy bone, and it's responsible for storing fat. There are two kinds of coverings on bone. We have the periosteum on the outside and endosteum lining the inside. The periosteum we find covering the outside of bones except in joint cavities. Where we have a joint, the periosteum becomes continuous with the connective tissue holding the bones in place. So if we have a synovial joint, the fibers of the periosteum become interwoven with the fibers of the joint capsule, and we'll look more closely at joints in the next chapter. The periosteum has two layers we mentioned before. It has an outer fibrous layer and an inner cellular layer. The functions of periosteum include isolating the bone from the surrounding tissue, providing a route for blood vessels and nerves, and also taking part in bone growth and repair. The fibers of our tendons get interwoven with the periosteum, and that's how our muscle attaches to our bones. So we would have, a muscle. This is a very rough drawing with a tendon coming down, and the fibers from that are going to interweave with the fibers into the periosteum. And then we can actually have fibers then that enter the bone itself, and those are called perforating fibers, or sometimes they're called Sharpie's fibers. And those are collagen fibers that get incorporated into the bony tissue from the tendons or ligament and the superficial periosteum. And that really allows for a very strong attachment of the tendons and ligaments to bone. Those fibers coming in get cemented into these circumferential lamellae. And so those fibers come in and they're basically stuck within those layers of the lamellae by the osteoblasts within that cellular layer of the periosteum, kind of cement them into there. So that provides a increased strength of attachment. Our other surface covering is the endosteum. And so we had the periosteum on the outside surface. The endosteum is going to line the inside surface of the bone. And the endosteum is actually an incomplete cellular layer that lines that medullary cavity and lines the central canals of compact bone and the trabeculae of spongy bone. It has this simple flat layer of osteogenic cells covering the bony matrix, and usually we don't have any connective tissue fibers involved here. There are areas where the cellular layer is incomplete and the matrix is actually exposed. And at these spots, osteoblasts and osteoclasts can remove or deposit the matrix components. The endosteum is active during bone growth, repair, and modeling because of this. Next up, we are going to learn about how bones grow. I'm a big Harry Potter fan, so I had to stick in a little golden retriever going to Hogwarts here. So some terminology. Ossification, also known as osteogenesis, is the formation of bone. Calcification is the deposition of calcium salts, which takes place during ossification, so it's part of ossification. But it can occur in other tissues sometimes due to disease or pathology. We can have pathological conditions for each of these processes, for ossification and for calcification. We have a disease called heterotrophic ossification, and that's abnormal bone formation in non-skeletal tissue. We can have ossification of muscles also as a result of genetic disease or trauma. Dystrophic calcification can happen in degenerated or necrotic tissues. As a reaction to tissue damage, we can get calcification or deposition of calcium salts within the tissues. And if we looked at this grossly or with the naked eye or on an x-ray, they appear identical. But if we looked at them histologically, if we took a little piece of this either ossified or calcified tissues and looked at them under the microscope and compared them, they are going to be distinct because one is actually bone formation, so it's going to have those osteocytes, the lamellae, etc., whereas the other one is just calcium salt, so it's not going to have that bone structure. There are two forms of ossification used by our body. We have endochondral ossification and intramembranous, and we're going to discuss both of those. Our bony skeleton starts forming about 6 weeks after fertilization, at which point the first bony elements present in the embryo are actually cartilage that will eventually be replaced by bone, and that replacement begins a few weeks later. Sorry, I've got hummingbirds buzzing behind me. After birth, the bones undergo a huge increase in size, and growth of the bones continues sometimes until about the age of 25 years. And this image on the right-hand side is just showing you what a fetal skeleton can look like with those the cartilaginous skeleton model that's going to be replaced by bone. The way most of our bones form is through a process called endochondral ossification. So as I mentioned before, during development, most of our bones start off as cartilage, and they start off as hyaline cartilage. Remember, we had a couple different kinds of cartilage we talked about in the tissues unit. Hyaline cartilage is where most of our bones begin, and they're like little miniature versions of the bones we see in adults. This cartilage is eventually replaced by bone through a process called endochondral ossification. And this process begins with a primary ossification center within the hyaline cartilage. And there is a video on the next slide that you can view that came with your textbook if you want to pause it, that goes through the steps. So if you want to watch this video, you can pause this and go to your PowerPoint for printing that has the link. So this endochondral ossification, we start in the embryo with that cartilage, that hyaline cartilage. And those cartilage cells, or chondrocytes, remember, near the center of this kind of shaft are going to enlarge. So we have chondrocytes within the center of this shaft of hyaline cartilage. They're going to enlarge, which basically makes holes in the matrix where they're enlarging. And then that matrix begins to calcify, and the chondrocytes begin to die and disintegrate. And that leaves behind holes within that now calcified matrix. So we've got these chondrocytes within that shaft. They start to grow, which is kind of pushing the matrix apart and making a big hole. The matrix starts to calcify around them, and then the chondrocyte dies. So then we're left with this big hole of calcified matrix behind. Then we have vessels that grow around the edges of the cartilage, and the cells of the perichondrium, so remember periosteum lines bony tissue, perichondrium is going to line the cartilage. Those cells of the perichondrium that line the cartilage convert to osteoblasts, and then that shaft of cartilage becomes covered in a superficial bone layer. So we get these blood vessels coming in, and the periosteum on the edge of that shaft, those cells convert to osteoblasts. Remember, osteoblasts are immature bone cells. And so we get this kind of superficial bony collar around that shaft. And then in the center, we've got these basically little holes within that calcified matrix. The blood vessels are then going to come in and enter the cartilage and go to the center of the shaft. Fibroblasts migrate with the vessels, and they differentiate into osteoblasts and start making spongy bone at what is now called the primary ossification center. So in the center of this shaft, we get what's called the primary ossification center. The formation of bone is then going to spread along the shaft toward either end. So it's going to go from either end. As growth continues, this structure is remodeling, and we get a medullary cavity in the center of the shaft. And the osseous tissue here along the sides of the shaft, so we've got that medullary cavity, and then the osseous tissue along the sides of the shaft becomes thicker and more solid. Remember, we're working toward making it into compact bone. The cartilage near the epiphysis is being replaced by bone as well. So with this step, we're getting an increase in length as well as diameter of the bone. Then we get some capillaries, and osteoblasts migrate into the epiphysis. So here's our epiphysis. We get capillaries and osteoblasts migrating into that epiphysis, and this creates our secondary ossification center, and we get these on either side of the diaphysis. Remember, diaphysis, epiphysis, metaphysis is where they meet. As this process continues, the epiphysis here becomes filled with spongy bone, and the metaphysis, located below it, the metaphysis is a narrow cartilaginous region, and we sometimes call it the epiphyseal cartilage or the epiphyseal plate, and it basically separates that epiphysis from the diaphysis. On the diaphysis side of the epiphyseal plate, and we can see kind of a close-up. Whoops, where's a close-up here. So the diaphysis side of the plate would be down here. On the diaphysis side, our osteoblasts are invading into this cartilage So the osteoblasts are invading the cartilage of the epiphyseal plate and replacing it with bone. And so that's replacing that cartilage in the epiphyseal plate with bone. On the epiphyseal side, so on this side, new cartilage is being formed at the same rate that the cartilage is being replaced with bone on the opposite side. While we're growing, while our bones are growing, this rate of production of new cartilage and conversion of cartilage to bone occurs at the same rate. So I just put this slide in here so I could draw. Normally in class I like to draw this on the board and so I can see in person, but here's our very rough bone, right? We've got our diaphysis, we've got our epiphysis, and then we've got our metaphysis. I was going to do that in a different color. Our metaphysis, where the epiphyseal plate is located. So while we are growing, from the diaphysis side, osteoblasts are converting this cartilage into bone, and on the epiphysis side, these chondrocytes are making new cartilage. So they're kind of chasing after each other. Osteoblasts making new bone, chondrocytes making new cartilage. And so the epiphyseal plate continues to be present and just kind of move along as the bone grows in length. At puberty, the rate of that epiphyseal cartilage production, so remember the cartilage was being produced here's our epiphyseal plate. So the rate of that cartilage being produced at the epiphyseal side, it slows down, while the rate of the osteoblast activity at the diaphysis side is increasing. And so what happens is we end up with that epiphyseal cartilage getting narrower and narrower until it eventually disappears. And what we have left behind is the epiphyseal line. And you can see that in this image here. This process is called epiphyseal closure. And you might hear people calling it closure of the growth plates. The only cartilage from that initial cartilage model, remember when we started this whole endochondral ossification, we had that hyaline cartilage model. So the only cartilage that remains from that initial model is the articular cartilage here that prevents damage of bones at the joints by providing cushioning. So the big takeaway from this, if we see an epiphyseal line, we're not going to experience any more growth in length. We can't, because that growth plate has closed, and that happens at puberty or throughout puberty. And so just once again, reiterate that point. Here's our bone, diaphysis, epiphysis. Here's our epiphyseal plate. So once we reach puberty, the production of cartilage at the epiphyseal side slows down, while the production of the activity of osteoblasts converting that cartilage into bone is increasing. In class, I like to kind of like pretend like we're having a little race, and the cartilage cells are slowing down and getting tired, while our osteoblasts converting into bone are speeding up until we catch up with each other. And then once we catch up, I wasn't supposed to erase that entire thing. Once we catch up, that cartilage disappears and we just end up with a line of bone called the epiphyseal line, and that indicates we are no longer going to have growth in length of the bone. So with that endochondral ossification process, we see both interstitial and appositional growth. Interstitial growth is a growth in length of the bone that we were just mentioning, and the formation of secondary ossification centers in the epiphysis is part of this process. And the continued lengthening that takes place at the growth plate or epiphyseal cartilage that we just mentioned is part of that process. Once that interstitial growth is done and we get that closure of the epiphyseal plate, we get that epiphyseal line, which can actually be seen on an X-ray throughout life. When in life that closure is going to take place depends on the bones that are involved, as well as the individual. If you took a series of successive X-rays of children growing, you can monitor the rate of closure by looking at how the cartilage narrows over time. And this image here is just showing you what it looks like. If we are still growing here, you can see there's kind of a radiolucent or black area here at the end of the bones, and that's where the cartilage is located. So this bone on the left-hand side is still growing. On the right-hand side, you no longer see that radiolucent or black area. Instead, you actually see a white line located in that location, and that is the epiphyseal line, which means that the growth plate has closed. We will have no more interstitial growth or growth in length. So in addition to that interstitial growth or growth in length in endochondral ossification, we also have appositional growth occurring. And this is a growth in the width of the bone. And it begins when we get that superficial bone or bony collar around the diaphysis in early endochondral ossification. So remember, we got that bony collar forming on either side of the diaphysis during that phase. We get the developing bone increasing in diameter at that outer surface, adding layers of those circumferential lamellae. So on a cellular level, the cells of the inner layer of that periosteum, remember the periosteum is lining the outside of the bone, and it had two layers. So if we had our bone here, the periosteum is on the outside and it has an inner cellular layer and an outer fibrous layer. So that inner cellular layer of that periosteum, those cells are differentiating into osteoblasts and depositing superficial layers of bone matrix to increase the diameter of the bone. Those osteoblasts eventually become surrounded in matrix completely, and then they differentiate into osteocytes. In the deeper layers, the circumferential lamellae get recycled and replaced by osteons that are typical of the compact bone. Remember, those osteons are the concentric lamellae that we talked about in previous slides. During this whole process, while those osteoblasts are adding matrix to the outside surface, osteoclasts are slowly removing matrix inside the medullary cavity, but at a slower rate than the osteoblasts. So we've got osteoblasts increasing the width. At the same time, we have some osteoclasts working inside, removing some of that bony matrix from the medullary, coming from inside the medullary cavity, but that rate of removal is slower than the rate of deposition on the other side. And this is how we get an enlargement of the medullary cavity while also getting an enlargement in the diameter of the bone. So we were just talking about endochondral ossification. Now we're going to talk about the other form, intramembranous ossification. Intramembranous ossification is also called dermal ossification because with this form of growth, the bone is developing in a mesenchymal or fibrous connective tissue, which is in the deeper layers of the dermis. Bones produced with this method include the flat bones of the skull, the mandible, and the clavicle or collarbone. And there are 5 main steps in this. So with this method of ossification, we see it begin around 8 weeks in the embryo, and we get these mesenchymal cells clustering together and differentiating into osteoblasts. They begin to secrete the organic part of the matrix. Remember, the osteoblasts in the matrix before it's calcified is that matrix before it gets calcified is called osteoid. The osteoid then gets mineralized with the calcium salts to form the bony matrix. So we've got, we've got our osteoblasts here. They're producing matrix here, whoops, which will initially be osteoid, and then it will become mineralized or calcified with those calcium salts to form the actual bone matrix. Where's my cursor? So those osteoblasts end up getting trapped in the bony matrix when and then they differentiate into osteocytes. The bone that is developing grows outwards from this ossification center in small struts, which are called spicules. So we're kind of growing out from here. And in this image, these would be our osteoblasts. And then in the center would be our osteocytes once they're surrounded by that bony matrix. We then can get blood vessels growing into the area between those spicules that formed, and this helps speed up our growth rate since it brings oxygen and nutrients to the areas. So we've got blood vessels coming in here in between these spicules that are forming. As the spicules grow, they interconnect, and this ends up trapping the blood vessels inside the bone matrix. So those osteoblasts continue their process of depositing bone with those blood vessels interwoven, and we end up with a plate of spongy bone. We get remodeling of the bone around the blood vessels, resulting in the spongy bone in the center, and then compact bone covering on either side. Remember, we had in our flat bones, we had that spongy bone sandwich. So compact bone on the sides with our spongy bone in the middle. The compact bone contains those osteons that we talked about, and we also end up with osteoblasts and connective tissue along the bony surface, which will become the periosteum. A little bit more about bony tissue and bones. So for our bone to grow and be maintained, we do need an extensive vascular supply. And because of this, osseous tissue is highly vascular. We usually see 3 major sets of vessels in larger bones, and these include the nutrient artery and vein. Most bones have only one, but a few, like the femur, might have more. And these vessels enter the bone through a round passageway or hole called a nutrient foramina found within the diaphysis, and you can see it here. You can actually sometimes visualize the nutrient foramina on an x-ray. You might take an x-ray of a bone. My very poor drawing skills. And when you look at the diaphysis, you might actually see a hole or a radiolucent spot within the diaphysis. And if you weren't aware that we had this nutrient foramina, you might be worried that there was a fracture there, but it's actually a normal structure because we do need to have a passageway for those blood vessels to enter the diaphysis. Branches coming off those nutrient vessels form our smaller perforating canals. Remember those perforating canals that go through the compact bone and extend along the length of the shaft into the osteons of the compact bone. Our metaphyseal vessels supply blood to the inner surface of the epiphyseal cartilage, right here. And this is where that cartilage is being replaced by bone when we're growing. And finally, we have periosteal vessels. These vessels are coming in from the periosteum, and they provide blood to the superficial osteons of the shaft. And you can see them in this kind of close-up image here. During endochondral ossification, branches of these vessels also enter the epiphysis and provide blood to the secondary ossification centers. After the epiphysis closes, we get that closure of the growth plate. These three sets of vessels all become interconnected. I just threw this image in here just to kind of cement in your mind what a great blood supply bones have. We actually can put catheters into bones. We call them intraosseous catheters, and we use this often in small animals that are just too small for us to get an IV catheter into. And this is also used in humans, sometimes in infants that are very tiny, or if a person came into, say, the emergency room and their blood pressure was very low and they were having a problem getting a catheter in. Sometimes if they needed quick access to the venous system to give a medication, they might actually put in an intraosseous catheter. We do also have lymphatics and nerves associated with the bones. That periosteum contains a network of lymphatic vessels that collect lymph from branches entering the bone, and those branches reach each osteon via those perforating canals. And then we have sensory nerves that penetrate that compact bone with our nutrient artery. This innervates the endosteum, the medullary cavity, and the epiphysis. And that is why when you injure your bones, it can be quite painful. I believe that is the end of this lecture.